It's my pleasure to introduce Dr. Kioumars Ghamkhar, a leader in plant genetic resources, phenomics, agritech, and agricultural innovation. Since 2014, he has directed the Margot Forde Genebank at AgResearch in New Zealand. With a PhD in plant molecular phylogeny and evolution from the University of New England in Australia, he possesses over 24 years of experience in plant genetic diversity, ex situ conservation, and cutting-edge agricultural technologies across Australia and New Zealand. He has pioneered the integration of omics data into GeneBank databases and has led award-winning research in plant phenomics. Kioumars also has entrepreneurial experience as the founder of Map&Zap, a revolutionary AI non-chemical weed technology that evolved into a startup. In 2023, Kioumars edited and co-authored a landmark book titled Plant Genetic Resources for the 21st Century, which focuses on omics and genetic resources. He serves as the co-chair of the academia section and is a board member of the International Plant Phenotyping Network, an organization aimed at integrating technology into plant phenotyping and promoting global standards in this field. The title of his talk today is Unchaining Genetic Diversity by Harnessing Omics Tools for GeneBank Innovation and Adaptive Agriculture. Welcome, Kioumars.
Thank you, Yanni, for the very generous introduction. And hello, everyone. Good morning, Europe, Middle East, Africa, if you're here. Good afternoon, Asia, or good evening. And good evening or late night, good evening to the Oceania region. And I have to say sweet dreams to my colleagues in the US, but I see some people have registered and Yanni is awake. So thank you very much, Yanni, again, for your commitment to this. Really appreciate that. So without further ado, let me share my screen. Unchaining genetic diversity for gene banks using omics technology. I want to give you a structure of the talk. I try to make it entertaining and informative, but it has different stages which I should give an introduction to first. So I talk about New Zealand agriculture and Margot Forde Genebank, then omics and the book that was introduced earlier. And then I talk about that, it's not just about the book, we walk the talk as well. What we talk about in the book, we actually try to practically operate on. Then we go to quite a few slides on seed phenomics and phenotyping, which Yanni was actually asking me to talk on that mainly, because I talked about this in Taiwan last year, April 2024. And then I introduced IPPN, International Plant Phenotyping Network, and collaboration with gene banks through that network. Germplasm phenomics is next, from ground-based to aerial and satellite, with some fun that I'll share with you. And then the cost value discussion, which is something Yanni again asked me to discuss before conclusion. So let's get started. So New Zealand is a country of 5.1 million people with about 268,000 square kilometers land. 50% of that is woodland, 39% pastures, 9% arable and horticulture, and about 800,000 hectares is irrigated. Agriculture R&D is government funded mostly, but also supported by levy-funded organizations, by farmers and government, and then private sector as well. 50% of our experts are dairy products, meat and fish, and then fruit and timber. So the focus here will be mostly on forages, but I've worked on on crops and grains as well. But most of what I present here is what I do at Margot Forde Genebank right now, which is forage species, for the reasons that you see here and also in the next slide. The exports that we do, it basically is, the contributions of 3 species to exports are high. One is perennial ryegrass, about $14 billion per annum contribution to the economy. Another is white clover mainly, about 3 billion, and more than white clover is pine trees, billion per annum. There are also a lot of other products that you see here. This is our exports in 2023 on the left. As you see, this area, whatever color that is basically focused on animal products, which is, the indirect income coming from forage species. We also have rough wood, and then fruits, wine, etc. This is well divided between different sectors, but the majority, as you see, is this, and we had $44.5 billion export in 2023. The point
Sorry,
on the
Kioumars, sorry, just a brief interruption. I'm so sorry to interrupt. I think the Zoom top part is blocking the full view. If you could just that the top part of your Zoom, and maybe
this one.
Yes, yes. Just so that we can see the title. The top of it is being blocked by the Zoom.
So
sometimes it can just be slid off to one side. See if that's possible. We didn't see it in the testing. Yeah, we still see it.
No. How about I share? I share again.
A quick suggestion could be, you see the circle on the right with the 3 dots?
Yep.
If you click on it, there is hide floating meeting controls. If you click on that, it will disappear.
A little bit further down. Little bit. Yes.
This one. Okay. Yeah. Oh, thank you.
Yeah,
thank
wonderful.
you.
Thank you so much. Sorry about that.
Thank you. We learned something tonight. I learned something tonight. So thank you. Now, on the right-hand side, you see the use of foreign crops around the world, or forages, whatever the country's economy is based on. And the heat map from white, basically, or very light green to dark green, is going from less dependent on foreign crops to really highly dependent. And as you can see, the new world is very much dependent on foreign crops, and many countries in the Middle East and North Africa are not, but Australia and New Zealand, of course, almost 100% dependent on imported crops and forage species. Now, the importance of genetic diversity, I actually put some grain and vegetables here, which are more attractive to show genetic diversity within a species. In forages, it's not as beautiful as that. Genetic diversity is the foundation of resilient and productive agriculture, as you know. This is what you see in a core collection of subterranean clover, which I developed in Australia about 10, 12 years ago, but you can still see the diversity. These are different types of subterranean clover which perform differently. Some of them more tolerant to stress, some of them less, but the shape and forms are different as well. But what is important here is that variation is essential not only for the survival and reproduction of populations, but also for the very existence of species diversity, because these, it's actually populations that end up creating new species. And we can see that in forages a lot, because forages evolution is way behind the crops in terms of domestication. This is on the left, what you see is a diversity in clovers. They got beautiful flowers and they have a huge diversity. There are 200 species of them. I just put a few there, so you can see the diversity between species. And of course, again, within species in clover and medics and all that, we don't have the diversity that you see in fruits, for example, in vegetables. Critical for climate adaptation, of course, this genetic diversity and pest resistance and yield improvement. We know all that, but important to remind people. Now, we all know that climate change and human footprint is having a negative impact on wild relatives. This is just an example of groundnut in the Amazonia region. Again, the heat map from yellow to red is the number of species. This is the current situation, and this is what is predicted to happen in 2055 on the right. So basically, you completely lose diversity. A lot of species will disappear, and probably 1 or 2 species will survive. We have the same situation for clovers in Southern Europe, this region. Basically what is happening is the climatic changes, the drought, the heat, as you can see in the map on the bottom, and North Africa and Southern Europe and Western part of the Middle East are where we're very interested in to collect more species, because these are extremely impacted by climate change and human actions, or basically the way that constructions are happening, or the habitats are being destroyed by humans. Now, the role of gene banks, of course, as suggested in these publications that I've just put a screenshot of them here, is to find and collect and preserve and provide access to plant genetic resources. And these dot points here are not the only roles. I know that many of you are aware of what we do at gene banks. Many of you do it actually, as I've tried to show in this picture on the left. We also support breeding programs, research and conservation efforts, and we initiate and introduce work on new crops and forages. This is very important. The last one, of course, all of them are important, but the last bit in the wake of climate change is quite important because, for example, as you can see, a lot of us, including forage scientists or forage gene banks, go to the regions that species are still surviving or new species exist that might be potentially new forages in the future. Tolerant to drought, tolerant to other stress, but also maybe having less output through animals, such as methane emissions, etc. And they are in regions that you cannot basically most of the time imagine, you know, in the side of the road or something. But then this end up being developed cultivars, as you know. So that's why we need collections. This is our champ, our seed hunter in New Zealand. His name is Zain, and he goes to Tajikistan, you know, Armenia, Russia, 2 years ago, Bosnia, and goes and collects these things. And as you know, there are 1,500 or 700 gene banks worldwide with about 7 million accessions. These are strategic global assets and safety net against the loss of valuable germplasm. So basically, these are priceless genes. We can't put a price on this. I was talking to Stefan Schmidt, who was visiting us last week from Crop Trust, about the value of gene banks. We have tried to put value on gene banks, and we have successfully done that, but it's still an underestimate for, you know, we've done it for our gene bank, and it's still an underestimate, and we think we have contributed about $5 billion to New Zealand economy through the gene bank in the past 20 years. So it's really priceless, and in a, you know, literally priceless, but also if we lose these genes, we can't replace them. So we have to do characterization, regeneration as well. It's not just go and collect these lines or populations or ecotypes, but we have to be able to characterize them, regenerate them, and maintain them, and hopefully distribute them to users as well. By the way, this is the Golan Heights between Syria and Israel. This is a place we visited in 2010. I think it's a very dangerous place these days, but that was a good picture we took. We didn't go to Israel, but took a picture there. So Margot Forde Genebank has got about more than 175,000 accessions, mostly forager species, but we also have the bread wheat, we have some carrot, we have quite a few other crops, let's say, or grains or vegetables, but mainly focused on forages. We got over 2,600 different species from 590 genera. These are sourced from over 120 different countries. We haven't gone to all those countries to collect, of course. Exchange between gene banks has been very successful. Within those seeds, that's the interesting part, within those seeds, we have more than 420 named fungal endophyte populations. That's also genetic diversity within genetic diversity. So those seeds, don't underestimate your seeds, I'm sure you don't, but the seeds are actually the habitat of this fungi or bacteria which might be useful in the future. In our case, in AgResearch, which is our umbrella organization for Margot Forde Genebank, we do a lot of research on endophyte, and these endophytes have created basically tolerance to drought, to disease in perennial ryegrass, for example. But 420 named fungal endophytes are there already, and a few, probably 3 or 4 times more than this number, unnamed or unidentified fungal endophytes. So you can see how important these are. Oldest population is from 1940 and is still germinating, which is a bit of surprise, but yes, it does. And I say surprise because in 1970s, when people started to basically run a kind of gene bank, not with with the current refrigeration system and things like that, but from 1970s they started to run this, and I was wondering if this 1940s is actually germinate, and they do. Over 8,000 wild populations have been collected in the last 5 years. So you see, we made it our mission to actually add and to it these numbers of accessions. And I think it's a very, very important mission to not forget about these days. The number one top priority for gene banks, I think the mission number one is to collect and preserve accessions. 3,300 accessions of ryegrass plus many other breeding lines are there. Many others like 30,000, and 2,500 accessions of white clover plus 20,000 breeding lines probably. So we have a huge collection of ryegrass and clover, but you know why, because I showed you the economic numbers before. So collections, this is the genesis, total number of accessions from genesis, and red is our collection, but a lot of our samples are hidden on the genesis collection, of course, because the number of accessions in genesis are higher than ours, but we now proudly share a lot of our data with genesis. We didn't up to 5 years ago, I think 5 or 6 years ago, but we made that our mission to make sure that we put more and more of our data on genesis. These are collecting missions in different countries. You can guess this is Russia, with a lot of vodka, people drinking vodka and going and collecting. So this guy feels hot there. But we also gone to Greece, and this is Bosnia that we just, I visited in 2022, and Zane went and collected material with them, and we have a project with them going now. So in brief, we need agri-diversification. We're intensifying trade discovery, efficiency for developing core collections, and then going from core collections to whole collections, and vice versa, and do fast characterizations. Fast characterization, I emphasize on this because we can't just have the same pace as before for characterization. I know some pioneering gene banks are already doing fast characterization, mainly genomics, through genomics tools, some metabolomics, but I think all omics should come in the in this story to make a very meaningful story at the end of the day. So without phenotyping, genomics can be kind of meaningless. So what I call computomics here is to put all these omics together so we can get an output faster. And of course, on top of that sits collaboration. And then, as we all know, investment is always not enough for gene banks, whether it's national gene bank or international. All of these matters are listed in the book that Yanni introduced to you. This was published in 2023, and I thank Crop Trust for endorsing this with an introduction. Both Mari Haga and Stefan Schmidt have put an introduction, and my colleagues, you'll see their chapters as we go in this talk. My colleagues from Crop Trust have contributed to this book as well. Now, why omics technologies? The chapter 3 of that book, Chris Richard from USDA and myself talk about this, but basically there are many reasons, but basically to enable deep insights into genetic potential of accessions. What is the, if we have 30,000 accessions or 300, what is the genetic potential of that group of accessions? The other is to accelerate breeding and conservation strategies, of course, through pre-breeding, through research that we can do with our colleagues or within the gene bank itself. We can accelerate identifying the right lines for future breeding programs. Now, what you see here is phenomics, which I'm specifically interested in these days because, you know, I've done genomics, metabolomics, all that, and it's time to really go and basically catch up with the speed and pace of genomics and metabolomics, etc., with phenotyping. The traditional phenotyping is quite a slow process and exhausting as well. So should go to that too. As you can see here, there are challenges in gene bank utilization. In a chapter in the book, my very good friend, who's now retired, Tony Brown, has talked about the evolving research themes in plant genetic resources, and he says where all these discussions started, let's say in 1980s or even earlier than that, and where we are today, what the questions are. But what is clear is the need for the use of technology or omics, data gaps and limited characterization. Most gene banks rely on traditional phenotyping. You see my very good colleague in Western Australia, Phil Nichols here in his young days when there was no gray hair, and other colleagues, again from Western Australia, doing traditional phenotyping, wonderful, but mostly semi-quantitative, not, you know, putting very accurate numbers on things, but it's basically 1 to 9, 1 to 8, 1 to 10, 1 to 5, and score that. And there's nothing wrong with that. It's just slow and it's not accurately quantitative. So slow integration into breeding programs is another issue. Challenges in translating genetic diversity into applied outcomes, and environmental uncertainty. This is the problem of the day, and climate change. We need better predictive tools for adaptive agriculture. So again, I know people have started doing that, but it's nowhere near enough, really. So we need phenomics tools, as you see here on the right, which is, you know, this one is about quality, this is about yield, to do these screenings faster and more frequently and quantifiable, basically with accurate numbers. So omics in action. What we do at Margot Forde is to boost our utilization using these omics technologies. Again, in the chapter in the book, we talk about genomics for germplasm improvement with Chris Richards from USDA. And we actually walk the talk, as I mentioned. There are publications, as you can see here. And this presentation, of course, is recorded. You can look at this later. But basically, the message is that whole genome sequencing for diversity mapping in subterranean clover is one example. We have a species that can be ideal for future of New Zealand agriculture or currently in Australia. And we have to make sure that we exploit all the tools to look at the diversities. We developed a core collection of 97 accessions out of the original 10,000 accessions or so. That was a long process, but now that we have that, we're really exploiting all that material. So whole genome sequencing has been done, but also we're doing pangenomics of within the clover group or genus, and resequencing 4 clover species. One of them is subclover. Subclover genome we published recently, this one here, but we're also doing the pangenomics, which is, you know, looking at resequencing the whole core collection. This is being done in 4 species, and then compared with other legumes as well, of course. Scheme sequencing of the same species is also being done to augment the pangenome. So this is by no means funded by GenBank. I emphasize this because some people might hear me and say, oh, this guy is coming from another planet. That's not the case. I know GenBanks don't have enough funding. So this is collaboration. As you can see here, JGI, Joint Genome Initiative in the US. We have a project with our colleagues in Sardi, South Australia, but also USDA, GenBank, and a few universities in the US, and we're doing this together. And also we do GBS. We have used GBS for developing core collections in the top 2 species of our interest. What you see here is the genetic diversity in ryegrass. It might not be very clear, but what it tells in the paper that I've shared here is that all the ryegrass that we have in New Zealand has mainly come from Spain and Portugal. But all these clades, or whatever you call them, groups that you see here, none of them have been really used or integrated with ryegrass gene pool in New Zealand cultivars. So we have quite a few cultivars of ryegrass here, but all of them are linked to Spanish Portuguese gene pool, which is quite risky because New Zealand has already experienced something not very good here, which is, you know, a disease hitting a crop and causing billions of damage. We don't want that to happen again. So you see, when humans look at something that works perfectly fine, which is a cultivar, which is highly productive, they don't want to move away from that. And that's not a good habit. We should change that. Metabolomics is another omics technology. Again, a few papers there, but I have a nice story here about functional insights into rumen methane mitigation in forages. We looked at forages and, ages ago with a student, and we threw a few species in there. One of those species came really as a surprise with very low methane emission from the animal gut. So subterranean clover, which is one of the low emission ones, was sitting up there, lower than red clover, and ryegrass is somewhere around here. And this species was sitting here. So what we did was that this is the core collection. After just one accession showed promise, we went to the core collection, looked at the core collection. Then we looked at the chemicals that, or biochemical components that might cause this. You can see the importance of this. If this is successful, it can change the game in the basically in the industry, because many people just blame the cows, of course, which are a problem for agriculture. But if something like this, which is a real feed, it's a forage, if you feed that to cows without any problem, then you can see what the consequences of that, good results of that could be actually. We have discussed the advances in metabolomics in research for germplasm improvement, and that's what, again, we walked the talk there and looked at the core collection of Bicerula, and then went beyond the core collection as well and looked at the bigger collection of Bicerula to see the diversity there. What is interesting, and we discovered here in New Zealand with my colleagues, is that even a third of that forage, if it's mixed with just normal forages like ryegrass, even a third of that mix, if it's this Bicerula species, then you still have about 70% reduction in methane emissions, which is amazing. So this is research coming out of Genebank. Now, omics in action again to boost our utilization. Another one is phenomics, my favorite one. We have a chapter there about improvement of crop adaptation. There are many things that you can do with this in phenomics, but mainly what we do here is on stress response. What you see here is using, for example, terahertz technology to look at early drought detection. Still in progress, and hasn't really finished. We haven't set it up, but it's new. I know a lot of people are working in terahertz, but we actually trying to do this in the field. So it's quite different to lab setups and glasshouse. We also work on forage quality. These are some variables in forage or factors that we like to measure in forage quality. And of course, total protein and nitrogen are important, and as well as some other factors, organic matter, etc. So for that, we use hyperspectral imaging, and we use combined and individual yield by species ID in forage mix, because in New Zealand, we use forage mixes so that the ryegrass, which doesn't fix nitrogen, is fed with a legume, which is white clover. So nitrogen fixation helps ryegrass to grow better. But you want to measure the yield by each component. Well, by yield here, I mean, because I'm talking forages, it's not seed yield, it's the vegetative yield or vegetative growth. But you can estimate the volume or the amount or the yield of each species separately using the combination of RGB imaging and hyperspectral imaging. What you see here is also just looking at basically growth. This is a fancy project that we did. Again, we don't have a lot of money. Sometimes somebody comes with a good funding, with an innovative idea. We have an innovative idea, they come and fund us, and we jump on it, and we put a range of germplasm in the field and look at them. So what you see here is the yield measurement, very accurately done. And also, in this case, you can also identify different species in the field. This is an aerial measurement system. Now, digitization turns gene banks to innovation hubs. That's actually our dream, but definitely my dream. My colleagues, thanks to Ben Killian, my good friend at Crop Trust, who's organized this chapter with Paul Shaw and others, they actually put a good chapter there about what can be done with data at gene banks, about how to combine things and get things talk to each other, like green global genesis, etc. But anyhow, what we need in the future is a digital gene bank. And at the moment, most of gene banks, all of us have a database. Of course, in 1980s, we had the 1980s style database. Very recently, about 5-6 years ago, we updated that to a very good searchable, Google style searchable, nice database called Seeds. And we can upload pictures, we can upload anything from the region that we collect, of course, maps of the region, and we use apps when we go for collection, and everything is automatically uploaded to the database. We need global initiatives to integrate omics data with germplasm repositories. And of course, as I said, we have the Seeds database, but we don't, we're quite open on moving to Green Global because of interoperability. We want to make sure that we are in line with all the gene banks or the system that is led by Crop Trust around the world. So again, as I mentioned, Genesis is, we're already participating and basically sharing our data on Genesis. So that is happening. But beyond that, just normal digitalization, which is, databases, we need to integrate AI and machine learning in gene bank management. Why is that? Because of this fancy picture. This is Tom Cruise from the Minority Report. I've always been fascinated by that movie and what might come in the future. I love to be able to put pictures together like him and predict what's coming in the future, but not for humans, but for plants. So I just put my own pictures here on this and our own sensors data. So integrated this picture together to say a gene bank should look like this in the future, to be able to do predictive modeling for optimal seed conservation and utilization, and also germplasm selection. Because right now it takes a long time to identify these promising lines. But in the future, hopefully with the omics technologies and fast scanning, we can have all this in a database like this, which is suggested by my colleagues in this chapter, and then hopefully make this a reality. I don't think we're far away from that, really. Now, impacts on adaptive agriculture, climate prediction, and climate match are important things that we should do. There's a chapter here, again, from a group of my colleagues, including Ben Killian, genomic approaches to using diversity for the adaptation of modern varieties of wheat and barley. But what we do at Margot Forde is that we look at what's happening in the areas that I mentioned earlier, Southern Europe, Northern Africa, and Western Middle East. And based on the current conditions, climatic conditions, we look at that climate match with what is going to happen in New Zealand in the next 30 to 50 years. And then we go and collect it in a targeted way. So, and it's also based on gap finding. So this paper published on Subtron and Clover a few years ago, for example, was telling the researchers where the gaps in the sub-clover collection is. I already mentioned there are 10,000 accessions of subterranean clover in the world. We have 97, a core collection of 97 accessions, but that's not the end of the story. There are still gaps. So you go and find the gaps, and also you want to avoid duplication as much as you can. But it's not just about gaps. There are also hotspots, which are, let's say, secondary centers of diversity that we should also go and collect again from those regions. So what we do in New Zealand is to identify those regions and go and collect. As you can see, northern and eastern parts of New Zealand, we see conditions that are similar to the current conditions in Southern Europe and North Africa and Western Middle East. And this is what we target for the future of agriculture in these regions. And the result of that is something like this. You see in Greece, you have a plant and type of clover, which looks nothing like a plant vegetatively, but the roots are amazing. On the other hand, white clover that contributes so much into our economy is actually having very shallow roots. So we have crossed, my colleagues here have actually crossed these two, the white clover and this clover, and they created this one. So this is the original clover that I said doesn't really have much vegetative parts, but it has really good roots, and they created this new hybrid, which is good for the future and for the dry areas. So basically, climate resilient crops and forages should be leveraged by gene banks and genetic resource centers. And then we can integrate omics and agroecological principles to create these opportunities. Now, this is the first part of the talk, and I know that we spent a lot of time already, so I tried to go through this part a bit faster. What you see here is the electromagnetic spectrum, because I want to talk about phenomics of seeds. And for phenomics, basically, we think that, some of these wavelengths are being already used in phenomics, and some aren't, or aren't as broadly used as others. But basically, we use this electromagnetic spectrum to convert data to images or information. This information you don't need, you can look at later, but this is basically the wavelength. And this one is my own categorization of seed traits. It might not match everyone's categorization or your standard categorization of traits, but this is what is important for me, for Margot Forde Genebank, as traits: physical quality, physiological quality, health quality, content quality, and of course genetic purity, which is important for everyone. But most of the things that I show here are not my publications, so most of the possibilities that I mentioned are not actually suggestions. They're already happening. Some of them are not, most of them are already happening. As you can see, all the references are here, and again, you can go and have a look at those. But let's go through them. Physical quality traits, such as seed being intact, can be looked at using RGB imaging. That's already a norm in some places. And there are publications in that field again. There are other traits about physical quality, such as uniformity, and by that I mean in size, shape, and color. Again, a lot of seed breeding companies use these technologies, these imaging ones, so it's quite a norm for them. But also multispectral imaging can be used for more accurate information. You don't necessarily need multispec for this kind of traits, but sometimes it's difficult, specifically if your seed size is normally small or the shape is not very diverse, then you might have to use other technology. Other physical quality traits include no contamination. So if you want to see contamination of weeds or other crops, again, RGB imaging can help. Multispec can help differently. RGB can help in terms of shape of the wheat seed from your normal seed, or maybe a bit of color, but multispec can help in terms of, multispec shows a different species. The seeds of different species have different reflection of multispectral waves. Also, thermal imaging can separate these things, crops, other crops from your crop or weeds from your crop, any contamination. Because again, depending on what species it is. The activity, physiological activity in the seed is different, so you can measure that with thermal imaging. And these are references to that. El Masri has done a very good job and review about this. If you want to read that, it's very good work. And hyperspectral imaging, of course, it's an expensive tool, but you can use multispec if, there are tools in the market which is not very cheap, but still you don't have to buy your own cameras. Some gene banks, including CG gene banks, I'm aware that they are buying these tools, the multispec tools for their screenings and characterization. There are also physiological quality traits like germination. Of course, RGB imaging has been in use for years for that trait, and terahertz imaging is another one. Terahertz, the good thing about terahertz is that it can actually show you the status of water within seed, and based on that, it will give you the potential of germination earlier than normal. You don't have to wait for the seeds to actually germinate. You can look at them at that earlier stage. Also thermal imaging, of course, because of the activities within the seed, the heat that it creates, the thermal imaging can detect that. Vigor, like longevity and stress tolerance, are other physiological quality traits which are again possible to measure using thermal imaging. And again, please look at these references here, because this is we don't have time to go to all details about this. I'm just quickly going. It's an overview of what can be done at gene banks potentially. I mean, thermal imaging doesn't have to be expensive. I mean, I've got a handheld one at the gene bank, and we can use that for this purpose, for example. Physiological quality traits or regeneration capacity, such as viability or reproducibility, can be looked at using RGB imaging. Of course, if you use different filters, normal RGB cannot do that, but it will look like multispec when you add those filters, but it's a bit cheaper than that. Again, the experts and the industry can help you when you go and ask for these tools. Thermal imaging can also help for the same reasons that I mentioned. All the activities that create heat, any signs of life or activity can be detected with thermal imaging. Of course, it depends what kind of camera you're using, and the cheaper cameras are less accurate than more expensive cameras. But again, this is a good paper to show you how this is done. Seed health traits. If you want to see if your seed is disease-free, you can use RGB imaging as in here in this picture on the right, or multispectral imaging on the left. Thermal imaging can also be used, and MRI, which is this one here, but that's too expensive for gene banks. I never use MRI, but people have done that in this paper. Comparative study has been done, but it's slow. For me, what is important for gene banks is fast phenotyping. So if it's slow and expensive, I wouldn't recommend it. So seed health traits, again, durability at storage, longevity we talked about before, but also hard seedness, which is, you see it in legumes a lot, or dormancy. And multispectral imaging can help with that. And this paper actually has done a very good job identifying hard seed from soft seeds using multispectral imaging in different species. So it's pretty good in legumes if you want to use that technology for fast screening of hard seedness. It depends how important it is for you. It's quite important for forage legumes that we work on, like clovers. For seed quality content traits, moisture and biochemical. For moisture, as I mentioned, terahertz measures water, so that's quite important. And then thermal imaging also can measure moisture to some extent, but it needs to have high sensitivity. If you use the thermal imager that I have, which is a handheld, not very expensive one, it won't detect moisture for you. Hyper and multispectral imaging can also be used, but again, that's more expensive. And again, multi-spec imaging is cheaper, and you can use that. But it's not always reflecting the wavelengths that you need. So hyperspec is more accurate, but of course more expensive. So biochemical contents, again, can be measured using hyperspectral imaging, which we are planning to do here. And another option for biochemical components is terahertz laser imaging, as per this paper here. But that would be quite, coordinating laser and terahertz is not easy. It needs a lot of programming. So again, I wouldn't recommend this for the seed banks. But if you're interested in the nutrition value of your seeds or things like that, of course, near infrared is available today. But it's destructive. If you want non-destructive, then you have to go for, further multispectral or hyperspectral technologies. So embryological and microbiome profile or embryological intactness can be done with X-ray. A lot of you are aware of that, I'm sure. It's already a norm. But also Wi-Fi is a technology that can be used with minimal damage to seed, but it's in development. It's not there yet, but watch the space with that. Microbiome MRI can help with that, but again, MRI is expensive, so multi-spec imaging can help if the right wavelengths are selected. This is one example of this work, which I showed before. And of course, genetic purity traits, GM versus non-GM material, can be looked at using terahertz imaging as per this paper just done. It is not possible in all the species, but some species you can do that. I'm sure some, modifications are needed for other species, but also for variety discrimination or cultivar discrimination, hyperspectral imaging can help. So it's not just the species that are different in their reflectance of hyperspectral wavelengths, but also within a species, sometimes not in all the species, you can detect different cultivars or varieties, as is done in this paper. Now, the last part of the talk, I talk about IPPN or International Plant Phenotyping Network. Yanni, please let me know if I'm talking too much. Just stop me when you have to.
We're good. Thank you.
Thank you. So International Plant Phenotyping Network is a network of academic and industry partners and private companies, which is trying to standardize phenotyping and make it affordable or accessible rather. But some of our network members, such as ULISH Institute and some CG centers led by Fiona Hay Studies and Fraunhofer, they have different technologies for seed quality testing or physical seed assessment. But again, most of these are really physical seed assessments. And what I defined as quality, I know what seed quality means by breeding companies, for example. It's exactly that. That's, the seed being intact and quality be good and embryo be not be damaged and things like that, but or uniformity. But to me, quality is also about what is inside biochemically. But a lot of our colleagues are already doing this in the seed space. So if you're interested, you can contact these partners as well. We have working groups in the IPPN, which I invite you to go and have a look on the website. But myself and Uli Scher from ULISH are leading the seed and germplasm phenotyping working group. And right here, I invite you all, if you're interested in seeds, which I think you are, being in this presentation, please do come and join us at IPPN, because we need more members here, because we want to, we don't want to reinvent the wheel. Let's put it that way. We want to discover things and standardize things together. So, and at that working group, we have challenges. Of course, challenges is to bring inexpensive, high throughput phenotyping in gene banks, and all these dot points here. I don't take you through all of that, but basically, as I mentioned, seed quality assessment, prioritization of crops. But what is important for us to have as an outcome is to suggest phenotyping solutions for gene banks and provide access to methodologies, and standard protocols, and also streamline use of technology and resources. So we think there has to be collaboration between gene banks and phenomics pioneers, or basically the organizations that are advanced in phenotyping. So gene banks don't have to do everything themselves, of course. And so far, we've taken a few steps. One was a workshop held in 2022 in Wageningen in Netherlands. And the outcome of that was a recent publication by a group of us, including 2 colleagues from Crop Trust. Hannes has left Crop Trust, I believe, but a very good colleague from Crop Trust, and Marlene, and Uli, of course, and Fiona Hay. And a follow-up workshop was held in Nebraska just in October. A follow-up to that, which was more about collaboration. We also published a review paper, which is not just about gene banks, but also talks about gene banks, which is called New Sensors and Data-Driven Approaches, a Path to Next Generation Phenomics. There are some suggestions there about what will happen in the future with the tech, and gene banks are mentioned as well. Now, more recent outputs and activities. We had a couple more publications on improving gene bank value and utility using phenomics with my good colleague Michael Selvaraj at CIAT, or Bioversity CIAT. And then upcoming 3rd workshop to be held in September 2025 in Horsham, Australia. There are 3 workshops. This is the main workshop there, which is, the longer workshop, which I've asked my colleagues in Australia, Sally Norton, the leader of Gene Bank in Australian Grain Gene Bank, will join me to run this workshop. It's a good place because we'll have a lot of seed scientists present. So at the International Plant Phenotyping Network, it's not all seed scientists, it's a lot of plant scientists, but here we have mainly seed scientists, so pretty good to run that one. So we invite you to join IPPN, but also if you can attend this conference. I want to promote this. I'm sitting on the organizing and program committee, and we're going to have a very, very good conference linking to seeds and gene banks. So if you can make it, you're welcome to join us. Still time to submit your abstracts. Just before I finish, I just quickly don't take you through all this because we don't have a lot of time. Just show you this general, germplasm screening. So it's not seed. What we've done is measuring growth, for example, in real time without harvesting plants. We measured growth every few days, and with a lot of plants, a lot of accessions. That's possible using LiDAR sensors, and we can now measure nitrogen to a good extent. Of course, it's not finished. The work is ongoing, but initial results show very good correlation between predicted and measured numbers. Remote sensing is another one that we've done. Again, don't do this at home. This one is different to what the gene bank's mission is. But I just tell you, when I say we have to be innovative and jump on opportunities, an opportunity came through, and so we got some funding from Callahan Innovation, and Airbus came on board. We could use their satellites to look at 10 farms, about 25 accessions of ryegrass, top accessions that we were looking at. So we asked 10 farms to grow 25 accessions each across the southern island of New Zealand. Well, Canterbury, let's say. That's a bit of an exaggeration to say the whole island. That was Canterbury. And then we had good funding, just 2 months project, and we could observe the growth and then the death of plants from satellite. But also we went on a helicopter and did some imaging from helicopter. We did ground truthing. So our troops, our field generals, let's say, were in the field cutting and harvesting and weighing. And we finally went on an aircraft. And I like to share a bit of fun of that with you if you're happy to look at this. see we put a sensor under the helicopter, quite risky, but we had a good health and safety procedure. So I stopped that here because you know it's going and going, but I edited that and I put Mission Impossible music on it for a reason, because it was really hard to do, but there was about 13 staff working day and night basically for about 2 months, and we were able to scan these accessions across farms in 2 months, and it helped a lot in our decisions. But again, this is, you know, this was just an exception. It's not the norm. But I just wanted to show the possibilities. Now, again, Yanni asked me to put some numbers in here. Don't take my numbers as a rule because I'm not the expert, but based on experience that we had, of course, omics technologies are expensive, and the cost and informativeness is different. But you can start with using basic molecular markers and do, you know, marker-assisted selection or whatever. But basic molecular markers for screening your material in GenBank. To go to GBS, that's what we use a lot at Margot Forde and our colleagues at AgResearch. Then there's transcriptomics, and the costs are right here, and that's about 200, you know, per sample, basically 200 to 1,500. That's US dollars. But again, this is an estimate, not 100% accurate. Whole genome sequencing, proteomics, and metabolomics, which is the most expensive. And right now, phenomics is about sitting in here. We're not talking about equipment, right? Because all these things need equipment, and this is just the operational and costs. But what we like to do for GenBanks is to move phenomics here. When phenomics moves down there, then I believe it's really useful for GenBanks, and people start using it in large scale. Right now, it's sitting up there, but when it comes down here, then it's more practical. And as to which phenomic technologies should GenBanks adopt, I wish I had Spock's tricorder. I don't know, for the younger generation, they probably don't know what I'm talking about, but this is something that could do everything at the same time and do the analysis inside the computer, the little computer, and then show you the results. That doesn't happen at this stage, unfortunately. And in that movie, from memory, I think it was 23rd century, when they were using this technology. But investment and adoption depends on which dimension you are interested on this graph. Again, this is an estimate, but I tried to put some costs here again. And this is about, you know, put 3 zeros in front of these numbers. And that's the cost of operation. So if you want to do high throughput measurements, you should go for RGB LiDAR technology. These are the cheaper ones, so you can measure more accessions. But if you want to measure more traits, then you have to go slower. If you want to measure a few traits at the same time, you have to go slower, and you have to go for more expensive technology. But if you want to do more accessions with less accuracy and just 1 or 2 traits max, then these two are okay. And this is basically the vehicle or the system that you should put your cameras on. Again, this is not a rule. You can have, for example, your terahertz on your robot, or you might have it on a platform, or things like that. At the moment, terahertz is on a platform, but we are trying to put it on a robot, for example. Or some people have X-ray on robots at the moment. But this is the general rule, not the so it can change. It can be different. So the take-home message is phenomics and genomics together, not alone, enable gene banks for core collection development, association mapping for traits of interest, with a special focus on climate adaptation for trait discovery, things like content quality and its transmissibility to the next generations, for standardization and consistency. I put repeatability in a bit of gray here because it's hard to get repeatability with phenomics. What is important is consistency. Because of different climatic conditions, you shouldn't really go too much with repeatability. Just for the final fun, I asked AI what the future gene bank should look like. And this is it. Congratulations, everyone. Your gene banks will look like this in whatever years from today. But I leave it to people to guess why it's next to the ocean. And then I asked, what would it look inside? Which I'm not sure why it put a human, silhouette there. But basically, these are the seed collections and plants growing inside. I think I didn't ask why it put it that way, but I think it's talking about advanced seed storage, automated robotic systems, AI-driven database and decision-making tools, and digital interfaces and smart tech, and bright and modern lighting. And with that, I'd like to thank everyone. I won't name anybody, but a lot of people have helped with genomics and metabolomics in this part, universities, research centers, gene banks, with phenomics, these people here, and with the aerial fun one, these guys here. Thank you very much for your time.
Thank you, Kioumars. That was excellent, the way you wove those many, many different topics together, quite complex by themselves. So I can imagine that the task of putting the last few graphs must have been a little bit daunting. We do have some questions, so I'd like to go into those right now. First question is, thank you, Kioumars. It always fascinating to listen to you. Can you please elaborate a little bit more on the methane production in Biserula, please?
Yes, thank you for asking. This Biserula palacinus is a monospecific genus, and just a serendipitous discovery about 2008, I guess, or 7. I had a student at University of Western Australia. I was living in Perth, Western Australia then, and I asked him to look at 13 species of forages, and with a colleague who's retired now. But in the last minute, I asked him to add this species as well, which was being grown in Western Australia and had 2 cultivars. So he did. And then when he came back, because he only tested 1 sample, I thought he's fallen asleep or something when he was measuring this one. And I said, how come this one is dropping methane by 90% almost? And he said, well, that's it. And then I said, well, you have to try core collection now. So we went and tested core collection against 2 of the lower, emitting species, and it came up again. So what is it? It's a forage that grows, that comes from southern Italy, basically, but it also grows in countries like Albania, which we're going to go to hopefully soon, thanks to my colleagues in Albania. And it's grown in Australia. So potentially, but it's not 100% positive. It's got some negative effects on animals. But the idea is to see what is causing this. So it's a forage. And when I said, yeah, the cows have been called the culprit, they might be, but it's not their fault. We can actually feed them with something and reduce the climate impact and still have a lot of good food.
Yeah, sounds good. Second question about Margot Forde Genebank. You mentioned 175,000 accessions, largely forages. How can you manage that? How many viability tests and regenerations do you do annually?
Thank you. Thank you for catching me. Not many.
Okay,
And
so
that's the
that's
problem because we are understaffed. That's a very good question. And the fact is that gene banks struggle with these things. I mean, if you look, I went to Vavilov Institute. I know Vavilov people are not here. We collaborated with them. I put them in acknowledgements in the past. Hopefully, we'll start working with them in the future. Right now, everything is disconnected from Russia. But regardless, so Vavilov had, I don't know, 30 staff or 25 people. I don't have that number. Stefan Schmidt came here and was surprised that we have three and a half full-time staff at Margot Forde. That's it. And the rest is casual. So they come and go and we pay them as we can, you know. So we can't afford screening everything. We just screen them when there is a demand for them or a request, or we are notified by the system that this thing is too old, like 1940s, and we have to look at them and regenerate it. But you can imagine with 3.5 people, we cannot really do all of that. But thank you for your question and catching me on that.
Yeah, no, well, that's a reality, right, of what we have. We try to save things and have longevity as a priority, but the staffing is always, yes.
Can I just add to this?
Sure.
Another reason I want AI to come to the system is that because AI can automatically notify you what is the priority to look at and increase and test for viability based on the data that you have in your system. That's what I'm trying to do at Margot Forde, you know, because I need it. So if we can have that AI to tell us when it's time for what accession to be checked, and you know, regenerated and things like that, that will be good. But basically at the moment is the demand-based and age-based.
Wonderful. Yeah, and you reduce regeneration, so that you're reducing a lot of the factors that, you know, identity loss and genetic integrity loss. So, I mean, there are advantages to not regenerating as much. We have one last question here. As you showed, analysis is associated with a certain cost. Gene banks are huge. What is your strategy to prioritize the selection of accessions to be analyzed? It's a kind of a part two question to what you said. You only have 3.5 staff. Given your constraints, how do you prioritize the selection of accessions to be analyzed?
Yeah, core collections. We do a lot of core collections. I mean, when I say a lot, I mean, we've done subterranean clover, we've done white clover, we've done ryegrass, we're doing red clover. So these species are core collection are being developed, but, you know, from say 3,300 accessions of ryegrass, we have reduced the numbers first to a 700 accession diversity panel, and then to 300 something. Again, by no means this is the final global core collection because we haven't looked at, for example, the polish material or things like that. But we have tried a lot of material from around the world. So with core collections, you can focus. And the studies that I showed you is all done on core collections, and then you can go back to the original. It's all about efficiency. Because with the lack of staff, the lack of funding, I mean, lack is not the right word. We don't have enough of that, you know, resources. Resources are never enough, so we have to learn to be efficient and fast and use technology, if that helps and answers your question.
Yes. We have one more question, if that's okay with you. Can we address that?
Absolutely okay. I'm
Okay,
100%
I find this a
energized.
very good question. I think for those of us that are starting on our way to using AI and other advanced tools, it said, what type of data should we store in our database for AI to make the decisions, for example, on viability? Like do you store a seed picture, or do you record water activity regularly? In other words, what data should be stored in a database so that AI can make a decision and monitor when you should be regenerating or alerting you at what activity should be taking place at a certain time?
First of all, I don't know the answer to that, to be clear. But then I give it a try. Because the reason is that this is quite new, right? We just at the moment the data that we have is just how the seeds look today. That's it. And if we're germinating them, how they look with germination, what is the percentage of germination? All this data can be, as an input for AI. But in the future, first of all, we need to decide collectively, and that's why we have that working group at IPPN. So I don't want to be, I don't have any right to be the sole decider on this. There has to be, so it has to be a global standard, right? What kind of information do we need in the future? But we have to prepare for this. So any data that we collect on the seeds with germination, with longevity, viability, shape, size, everything should all be in the database and then fed into the AI, because our brains cannot process those data all the time. And again, that's why the databases are so important. But the database alone is not enough. The AI program should be, deciding based on all these factors and see which seed is the priority to measure. Again, this is a rough idea. I don't have the answer and solution for this, but thank you for asking, and I ask for your help as well in that space.
Yeah. I think that's a fantastic question, is what would the formula be like for AI to be able to read this, from moisture content to oxygen in the bag to longevity and intrinsic things within each species that make it last longer or not?
Absolutely. Again, going back to what I showed with hyperspectral imaging or multispec, or the tools that are in the market already, you can buy, they're not very cheap, but or just even thermal, even, whatever. I have a terahertz camera here now. Maybe I use that for the seeds. So anything that we can measure and use and enter the database can be information for AI and enrich the AI, because AI, as you know, machine learning gets smarter and smarter as it goes, and eventually turns into deep learning by learning more and more information, and then it's smart, and then it's smarter than us eventually. But I mean, in terms of fast processing. But yeah, basically, I would say anything that we can should feed this database with, until we decide what is good, what is not, and it has to be a collective decision.
Thank you. That was wonderful. So I think with this, we'll bring the Grow webinar to a close. Thank you, Kioumars. As always, a very fascinating and thorough presentation. Thank you so much for taking the time to educate us. To Faye Wambua-Ludeling behind the scenes, thank you so much. And of course, to you, the audience, thank you for attending and staying on beyond the hour. Kioumars, any last words of goodbye to you?
Just wanted to thank you all and say, let's become best in our plant biology first, and genetic diversity, understanding diversity and plant biology, which we do, I'm sure, but basically master in that first, and then go to technology. But don't delay technology too much, because the world is changing, and gene banks should be innovation hubs. We can make it that way, and not storage places anymore. And thank you for your time. I apologize that my time zone is different to everyone else, and specifically to Yanni and anyone in the US or Canada. If they are here, I apologize.
Thank you again. Thank you, everybody. See you in the next Grow webinar. Bye-bye.
Thank you. Bye-bye.